Well, talking about the topics of the vitamins, we're quickly going through what we have gone through till now. That the vitamins of two groups divided into two: water-soluble vitamins and fat-soluble vitamins. We have gone through the fat-soluble vitamins, vitamin A, vitamin D, vitamin K, vitamin E, and we have gone in detail of the vitamins, and at the end I would also let you know the university question regarding the vitamins, very important. And coming to the water-soluble vitamins, the water-soluble vitamins divided into two groups. One is the B complex group and the other is non-B complex group. And you are now familiar that there is only one vitamin which falls under the heading of non-B complex, that is the vitamin C, and it is also known as ascorbic acid. Whereas coming to the B complex, there are a number of vitamins in that. If we're talking the energy releasing vitamins, that come to B1, that is thiamine, then vitamin B2, the riboflavin, vitamin B3, niacin, then is the biotin and pantothenic acid. They fall under B complex vitamins, but they fall under the group of energy releasing. Then under the heading of B complex, there are two vitamins which are the hemopoietic, that is the folic acid, and vitamin B12. There are other vitamins also, that is the vitamin B6, pyridoxine, and that is coming in under other groups. But what today my discussion is, that is regarding the vitamin B12, that is the cobalamin. Before going into the sources and other things of this vitamin, I would like to stress, would like to tell you a very important thing, that why this vitamin is important. I was talking about vitamin B12, that is the cobalamin. First of all, I think it's important to let you know why this vitamin is important and where this vitamin is taking very important participation in reactions of the metabolism. So that is very important. So vitamin B12, it is required in humans for two essential enzymatic reaction that is shown in this diagram. One is the first one at the top, that is under the mark of A, that in this diagram it is the remethylation of the homocysteine. Here, the redly marked, it is the recyclation of the remethylation of the homocysteine to methionine. It is forming methionine. And you see here the vitamin B12 is importantly used, it is involved, otherwise it will be not converted into the methionine. The second one is the coming into the diagram B here, it is the isomerization of methylmalonyl CoA, isomerization of methyl, that is methylmalonyl CoA, which is produced during the degradation of some amino acids. What those amino acids? That is isoleucine, valine, then is threonine and methionine, and fatty acids with odd number of carbon atoms. Here again, this is involved, B12 is involved. When cobalamin is deficient, now what's the importance here? I have told you these two reactions. What is the importance? When cobalamin is deficient, unusual branched fatty acid accumulates and become incorporated into the cell membrane. That will be not proceeding, and these amino acids will be incorporated into the cell membrane. Which cell membrane? Those of the central nervous system. So this may account for some of the neurological manifestations of vitamin B12. So that's what's very important. Now, what I was talking while telling you that some odd branch chain amino acid, and on the diagram of A there is the formation of the methionine. Let me show you the part of that map. What I talked to in my very first lecture, that always keep this map in your VN, go through this map very rightly. This map is now very clearly indicating that if you look here, the odd chain, that is acids, fatty acids, they are converted into converting into the propanol CoA, then methanol CoA, and then ultimately into the succinyl CoA. Here is this diagram. And I told you that isoleucine, methionine, valine, they are again converted into the propanol CoA, and propanol CoA, then methanol CoA, and methanol CoA, then succinyl CoA. And you see, succinyl CoA is very important member of the TCA cycle. So now, telling you all this background, what I want to stress is that the importance of vitamin B12, water-soluble vitamin B12, that is the cobalamin, that if there will be deficiency of this vitamin, it will be not taking place, the homocysteine will be not converted to methionine, and that methionine then ultimately converted into the succinyl CoA, and the second, as that is the methanol CoA, that will be not converted into succinyl CoA, and what will happen? Accumulation will be taking place, and as I told you, that then the involvement of the central nervous system. Now the detailed view of the structure of vitamin B12, that is the cyanocobalamin. Now I call it, previously I talked to you about cobalamin. Now I am adding another word, that is the cyanocobalamin. If you look at this structure, it's very easy to understand the structure. That is there is a central portion of molecules consisting of 4 reduced and extensively substituted the pyrrole rings surrounding a single cobalt here. These are 4 rings. This is being shown with this indicator that it is cobalt here, and it is being surrounded by these rings, that is the pyrrole rings, and this system is called the 4-ring ring system. So talking about the structure of vitamin B12, I am telling that there is a center which is the cobalt atom, and that is surrounded by 4 reduced and extensively substituted pyrrole ring. And this system here with the cobalt and fourth, that is ring, is known as the chlorine ring system. Now coming below this chlorine ring system is the DBI. Here it is the DBI ring, that is 5,6 dimethyl benzoyl ribose. It is here, that is here, it is attached, this whole DBI ring, that is which is in this ring there is the 5,6 dimethyl benzamidazole riboside, which is connected at one end here with the cobalt. So two things now are discussed. One is the first is the chlorine ring here, where is the cobalt present, and that cobalt is surrounded by four rings, that is the pyrrole rings, and this cobalt is then attached and is connected with another ring, that is the DBI ring, and that is what is known as the dimethyl benzamidazole riboside, which is connected with the cobalt. Then if you, after these two look, here at the top it is also connected to the cyanide group. The cobalt, on one hand it is connected with the DBI ring, and on the other hand connected with the cyanide group, is bonded to the cobalt atom, and that is why it is known as cyanocobalamin. Now you can well understand why we call it cyanocobalamin, because there is attachment of the cyanide group. If that cyanide is removed, then this will be called as cobalamin. If the cyanide is removed, this will be known as the cobalamin. It is known as cyanocobalamin because cyanide group is attached with the cobalt group. There are certain isomerization and other forms of the vitamin B12 also I would like to share for knowledge sake with you, that if in case of cyanide here on the top, if there is a substitution of the hydroxyl group, that is the OH group, that form of vitamin will be known as hydroxycobalamin, because OH is hydroxyl group, then that will be known as the hydroxycobalamin, and we call it vitamin B12A. This is a detailed division of the vitamin B12, that is the hydroxycobalamin. Why it is known as hydroxycobalamin? Because at the place of cyanide attachment, which is attached to the cobalt, there is, if that is replaced by the OH group, that is the hydroxycobalamin. Okay, another good thing, that if this cyanide group is replaced with the nitro group, NO2 group, instead of here at cyanide, if NO2 is substituting or coming, then this will be known as the nitrocobalamin, and that is given the name of vitamin B12C. Here is, in the case of hydroxycobalamin, B12A. In case of the vitamin where the nitrocobalamin is attached at place of cyanide, it will be known as B12C. Another option, if chloride Cl group is replacing cyanide, then it will be known as chlorocobalamin. And another possibility, if the SO4 sulfate group is replacing cyanide, then it will be known as the sulfocobalamin. So I have told you that the vitamin B12, it will be further, can be going into different formats. This classification, different forms, and those forms are the vitamin B12A, vitamin B12C, and that is cobalamin and sulfocobalamin. Why this name is there? That when cyanide group is replaced by OH group, chloride group, sulfate group, or NO2 group. Biologically, action of these derivatives, what I told you, are similar to cobalamin, but I stress it again, the biologic actions of these derivatives, what I told you, that is B12A, B12C, they are similar to cobalamin, but hydroxycobalamin, that is vitamin B12A, is superior as it is more active in the enzyme system. We'll be talking about the metabolism and absorption and excretion of this vitamin. That vitamin B12 is absorbed from the ileum for its proper absorption. It requires. Now, before this diagram, I would like to show you. Okay, let's talk from this diagram, and then we'll show you that written form. If you look here, it's the diagram here. It's self-explaining and very clearly indicating that in the diet that is available coming to the stomach, vitamin B12, and then this vitamin B12, I'm marking it here red. You can see. Very carefully look into this diagram here, and then there is in the stomach coming, there is HCl is also present there, which is important for the action of this vitamin also, and then there is a protein involvement, and intrinsic factor is very important to proceed this vitamin B12 further. What is happening? You can look here that the vitamin B12 is joining with the intrinsic factor which is produced by the cell wall, and that intrinsic factor is very important. If that intrinsic factor is not there, and in certain diseases there is deficiency and intrinsic factor is not there, then certain diseases can take place, which I will let you know later on. But here only I am telling you the movement of vitamin B12, how it is absorbed, and in combination with the intrinsic factor, it will be coming to the ileum, and here coming again, then what will happen, that the intrinsic factor will be retained, whereas the vitamin B12 will go into the blood, and that will be in combination with the certain protein there. So one very clear indication is that now I thought that what I taught you is that for the absorption of this vitamin B12, presence of HCl is important, number one. Then the presence of intrinsic factor, that is also known as IF of Kessel, a constituent of normal gastric juice, and this intrinsic factor IF, what I told you, it is secreted. Now it is written here, what I am showing you, that this intrinsic factor is secreted by the parietal cell. What is this IF? It is glycoprotein, a constitution of the gastric mucoproteins. I repeated that the intrinsic factor is a glycoprotein, a constitution of the gastric mucoprotein. In addition to amino acid, there are hexoses, hexamine, and it is non-dialyzable, and it is also thermolyable, destroyed by heating at 70 to 80 degrees centigrade for half an hour. So this is what is the intrinsic factor. If there is atrophy of the fundus of the stomach and a lack of free HCl, when HCl is deficient, there is a lack of HCl, it will be known as the aclohydria. This is usually associated with pernicious anemia. So this is what I want to stress, this is what I want to tell you, that in case of the deficiency of vitamin B12 and how this deficiency will be occurring, then in that connection I told you that there is the atrophy of the fundus of stomach, then there is a lack of free HCl, that is aclohydria, and that condition then is associated with the disease, the problem, and that is the pernicious anemia, and it is caused by, we can call it, it is caused by the B12 deficiency. Though it is due to deficiency of B12, but why this deficiency developed? Because there was, if there is a intrinsic factor is not present, if intrinsic factor is defective, if HCl is not present, then the pernicious anemia will take place. Now, when I was talking of the gastric juice, the gastric acid and pepsin releases with vitamin B12 from protein, binding a food and make it available to bind to the salivary protein, that is the Cobalamin. It is duodenum that is hydrolyzed, releasing the vitamin for binding to the intrinsic factor. There is another good diagram to understand the absorption, the mechanism of absorption of B12. This diagram is, I'm showing another one, similar what I showed you in the case of stomach diagram. Here I'm indicating again. If you look at the top of this diagram, there is the dietary food containing vitamin B12. You see, I talked that HCl is important, is needed. HCl is pepsin in stomach, that's good. Then saliva is also having the cobalamin salivary protein, and these protein coming in combination with the food and vitamin B12. Here at this junction you can see that, and then is the duodenum, and then it is moving further, then binding of vitamin B12 with the intrinsic factor. IF is the intrinsic factor. The next one, in the next phase, the attachment of intrinsic factor B12 complex to the receptor gene. Here is what I taught you, and then that is the ileal mucosal cell you have seen here in this, and B12 is released in presence of calcium ions and a releasing factor. Then it is released. Now this B12 IF is detached here. First, the intrinsic factor attached with the vitamin. Now, this intrinsic factor is detaching. This is detached, probably enters lumen of the ileum, absorbing into cell, mucosal cell, and ultimately the binding of vitamin B12 to the transcobalamin if it is in portal system, carried to the liver for the storage. This is how this vitamin B12, through the help of the intrinsic factor, in the presence of HCl and presence protein, and the help of the salivary protein, then is moving, and ultimately it is stored in the liver. talking about the clinical aspects. In pancreatic inefficiency, the cobalamin bound vitamin B12 may not be split and the complex is excreted in feces, resulting to development of the vitamin B12 deficiency. Now you see, vitamin B12 is absorbed from the distal third of the ileum via specific binding sites, receptors that bind the B12 IF complex. The removal of vitamin B12 from the intrinsic factor in presence of calcium ion and a relieving factor secreted by the duodenum take place and B12 enter the ileum mucosal cell for absorption into the circulation. And if you see the ileic absorptive mechanism is functioning, it can adequately transport that 0.5 to 10 micrograms of vitamin B12. A small portion amount, about 1 to 3%, may be absorbed by simple diffusion mechanism also.